Hi, my name is Suresh, and I'm from the Brain Stimulation, Imaging, and Cognition Group. And today I'll be giving a talk on the rapid timing dependent increases in corticospinal excitability during paired pulse TMS. Okay, so TMS is a non-invasive brain stimulation methodology that utilizes a magnetic pulse to change structures in the brain, which alters behavior and cognition. One variation of TMS is RTMS, which is repetitive TMS, where multiple pulses are applied to the brain to increase changes in the brain, as well as induce long-lasting changes of brain excitability by enhancing neuroplasticity. One way to test this is to use a combination of TMS and electromyography over the motor cortex and measure motor evoked potential at the hand muscle, using them as a proxy of cortical changes. And by indexing these changes in MEPs, we can then determine the efficacy of stimulation paradigms. So one of the problems right now with RTMS is its utility is currently limited due to its high degree of individual variability. And this can be seen in a study shown by Hamada and colleagues, where they used a very well-defined stimulation paradigm, theta burst stimulation. And when it was applied to a bigger population of 50 people, they saw that there was a huge degree of variability as well as no net change in the bigger picture across the entire stimulation paradigm. And as a result, the key to harnessing the potential of TMS would be to understand more fully the factors that contribute to the variability, and this will help us design better paradigms. Right now, the challenge in designing paradigms is that there's a scalability issue, and where the multiplication of the various parameters to designing the paradigm makes it experimentally impossible to test them all. For example, if we were to take three parameters, such as number of trials, the intensity, and the interstimulus interval, and let it vary by 5 degrees, we would end up having 125 potential variations, and that would not be possible to test all of them. As a result, a systematic approach has to be taken in order to determine which combination of variables would yield the optimal results. One way to do this is through TMS physiology, and this is an approach that Dick Bloom and colleagues took when they incorporated concepts of eye wave physiology to design their stimulation protocol. Now, eye waves are indirect projections from cortical interneurons onto the corticospinal neuron, and they choose to use a paired pulse TMS paradigm because when a paired pulse is applied to a time of net excitability, this then increases the chances of facilitation. As a result, they chose to use an ISI of 1.5 milliseconds, as there was a net excitability which coincided temporally with the descending volleys of the eye waves. What they found from their results is that throughout the TMS intervention, there was a near to linear increase in MEP amplitude, finishing with a 500% increase compared to the pre-stimulation MEP. But more importantly, there were long-lasting effects lasting for 10 minutes post-stimulation protocol. Next. However, eye wave is not the only temporal point of net excitability. There are other points of net excitability located later in time, such as the intercortical facilitation protocol that usually occurs at 10 to 30 milliseconds. Typically, this protocol is run using a subthreshold conditioning pulse. However, a study by Biabani and colleagues showed that a suprathreshold conditioning pulse caused a larger amplitude at the targeted temporal region, followed by a net inhibition. As a result, we would like to determine how and if MEP amplitudes change within the stimulation blocks resulting from this suprathreshold conditioning pulse in a paired pulse paradigm by indexing the MEPs during stimulation blocks at various ISIs. We plan to use data from a previously published study in the Journal of Physiology by Biadabani and colleagues that aimed to determine whether EMG and EEG measured the same, index the same cortical responses. And this study utilized the paired pulse paradigm. Some of the specifications of this data that's being used is there were 16 people and all of them were right-handed and healthy. So this study was conducted by 8 blocks, where each block was comprised of 20 trials and had an inter-trial interval of 5 seconds. They used a conditioning pulse of 120% of the resting motor threshold, and 1 millivolt was used for the testing pulse. Each block varied in ISI, where there were ISIs used from 10 to 200 milliseconds, and a general linear regression model was used to analyze the data. What was found was we did a comparison between the main effects of the interstimulus interval and the trials, and there were interactions between the two, and we found that there were effects of the interstimulus interval as from variations on the rate of plasticity induced by the paired pulse paradigm. And what we found was there were significant changes at 10 milliseconds, 20, 30, 40, and 200 milliseconds. And what these data points represent is the average change of amplitude between trials. And what you can see at the highest point at the 20 millisecond mark, you can see a significant and rapid increase of amplitude throughout the trial, where at a non-significant mark, such as 150 milliseconds, there's almost no net change throughout the entire trial. And what we can deduce from this is that the suprathreshold paired pulse stimulation results in rapid increases in corticospinal excitability at certain ISIs, especially the 20 millisecond ISI. From these future directions, we'd like to build on these findings by determining whether these effects last longer past the stimulation protocol. We plan to do this by indexing the MEP post-stimulation, and whether these changes have cortical contributions. We plan to address this by running a TMS EEG study. I would like to thank my supervisory team for helping me prepare for this presentation, and thank you for listening to my talk.